we're going to just take a quick look through the male reproductive system here. In class, we already began taking a look at it, but I'm going to run through it one more time so that ensure that you have it all for your notes. So we started just by looking at the anatomy of the male reproductive system, recalling that primarily you'll be asked anatomical questions in lab. So although we're certainly going to be looking at the function of the cells of the testes and the formation of sperm, and you'll do need to have at least a basic understanding of that flow of semen, where it's formed with the sperm, the fluids from the seminal vesicle, the fluids from the prostate gland, and the fluids from the bulbourethral or Cowper's gland. So you would need to be familiar with those fluids, although that's primarily content that's going to come up in lab. Looking at the layout, I always like to show this picture to remind everyone that when you're looking at a mid-sagittal view, that you sometimes look like, oh, there's only one of two testicles, but only one set of tubes. But no, in fact, there are two sets of ductus deferens coming around. They join together for the ejaculatory duct as they work their way in through into the urethra. So I'd like to have you see this picture. We start by looking at the scrotum. That's that two-chamber sac that holds the testes. Here's our picture of it, showing those two testicles within the muscles of the scrotal sac. There were two muscles that I asked you to know. They're on that previous slide, the cremaster muscle and the dartus muscle. The cremaster muscle is skeletal muscle. It extends from the abdominal wall into the scrotal sac. And the dartus muscle is the muscle, that smooth muscle that lines the scrotum. So the dartus muscle is lining the scrotal sac, and the cremaster muscle is extending down from the abdominal wall into the scrotum. Recall that the function of those two muscles is to help regulate the temperature within the testes, because sperm development requires the temperatures of about 95-96 degrees Fahrenheit, which is below normal body temperature. I also mentioned in class the testes are homologous, or basically of the same type of tissues as the ovaries. The difference being that egg production does require a higher temperature, and the ovaries stay up within the pelvic cavity in the female, whereas these testes will descend down the inguinal canal during about the 7th month of development, of fetal development, and extend into the scrotal sac. So initial development is up within the pelvic cavity, but they will descend down upon development. Okay. So those are the two muscles that you need to know there. Epididymis and ductus deferens in terms of their function. The primary function I want you to be thinking about with the epididymis is that it is the location for the maturation of the sperm or spermatozoa. There is some phagocytosis that goes on, and there is some fluids that are being monitored and regulated through the epididymis, but its primary function is the site of maturation for sperm cells. Sperm are made in the testes, and they will travel to the epididymis, where they will remain for up to several weeks to complete their maturation process. And then the ductus deferens, that tube that's lined with smooth muscle, is also a place for storage of sperm. So there's not any real maturation in the ductus deferens, but it is a storage site. Then looking a little bit more closely at the testes themselves, I mentioned they were paired. I mentioned that they'll descend in that inguinal canal around the 7th month. The testes is actually divided into lobules. And I'm going to jump to the next picture here for a second so that you can see these lobules that are here. It's actually blocked off right here. So you've got these lobules right here. Within each lobule is 1 to 3 seminiferous tubules. Right? So when we look here, you can see they're just showing you really one in the picture, but these are the seminiferous tubules. They're labeled right in here. So each lobule has 1 to 3 tubules inside of it. Inside the seminiferous tubules are where the sperm cells are actually functioning, where they're made. And then there are also what are referred to as nurse cells, Sertoli cells, or sustenacular cells, and all three of the terms are interchangeable. I tend to use nurse cells or Sertoli cells more commonly, so that's the terminology I'll use on the exam, nurse/Sertoli cells. I tend to not use sustenacular. I think it's an older term. But those cells are going to provide some very specific functions to help with the sperm development. We'll look at those in just a second. And then surrounding the seminiferous tubules, still inside the lobule, but not inside the tubule. So if I go back up here, we don't have a really great picture of it, but in this kind of pink area, so you've got the lobule, you've got the seminiferous tubule, and then in the pink area surrounding it, that's where you will find the interstitial or Leydig cells, and I use those terms interchangeably. And those cells surround the seminiferous tubule, and you will see in just a moment that that's where testosterone is specifically made. So sperm cells and support or nurse or Sertoli cells are inside the tubule, and then surrounding the tubule are the interstitial or Leydig cells. So what do those nurse cells do? Well, the first thing they do is form tight junctions that make the blood-testes barrier. Now, hopefully you are familiar with the term blood-brain barrier. The blood-brain barrier is formed by specific cells, epididymal cells, and cells within the brain that prevent substances from entering into the neurons of the brain. So that's part of why sometimes it is extremely difficult to treat certain brain diseases, brain cancers, because the blood-brain barrier from those neuroglial cells, such as epididymal cells, they are such tightly formed junctions that things cannot diffuse or pass through those cells. So that protects brain tissue. In the case of these tight junctions that we see in the blood-testes barrier, it's a little bit different. In this case, what we're trying to keep out is actually our own immune system. If you'll recall, meiosis, which we're going to look at in just a minute, in the meiotic process or meiosis, we go from diploid cells where we had 46 chromosomes or 23 pairs to haploid cells where we have just a half. We separate those pairs of chromosomes, and so instead of having 46 or 23 pairs, we only have 23 individual chromosomes. We've separated the pairs. That's part of what has to happen in meiosis, so that therefore that germ cell can be joined with another cell from the female, in this case if we're talking about the male. So you can't bring together two sets of 46 chromosomes. You will not form a human being. You'll form some massive cluster of cells that cannot develop into anything. So meiosis is very important to split the chromosomes, to split the genes, so that you give half from each male and female. However, when your cells of your body have an abnormal number of chromosomes, and anything below 46 or 23 pair is considered abnormal, your immune system says, hey, that's a mutation, that's a problem, and I'm going to destroy it. Well, in the case here, this of course isn't abnormal, because we're talking about germ cells, gametes, forming this. However, your immune system can't really tell the difference. It sees it as abnormal and wants to destroy it. So the blood-testes barrier prevents your own immune system from destroying, in this case obviously in the male, from destroying those haploid cells. So that's a very important function. These nurse or Sertoli cells ensure that the haploid cells can continue on with their development. These cells also interact with testosterone and FSH to promote the mitosis and meiosis. If you'll recall in class, I talked about the synergistic effect of two hormones working together, and I gave the example of T3 and T4 and the epinephrine having very strong synergistic effect. Well, the same is really true here with testosterone and FSH. You need both of them to work together to promote the formation of sperm, and you need those nurse cells to provide kind of that landing spot for those hormones. These cells also nourish the spermatocyte, spermatid, sperm. Those are the developmental levels. They're basically all forms of the sperm. They clean things up. They phagocytize things. They secrete a hormone themselves called inhibin, which has a negative feedback on FSH, so it keeps FSH in check. And it controls the release of the spermatozoon, or the nearly developed sperm, into the lumen so it can go into the epididymis next for its final maturation. So these are important functions. Expect question on the exam to ask you to discuss several of these functions. I've got 6 listed here. I ask you usually to give me 4 of them and describe them. And then, as I mentioned earlier, the interstitial or Leydig cells actually secrete testosterone. So that is now, by the way, your site of secretion, site of synthesis and secretion of testosterone. Before we were just going to say the testes or the gonads. Now you have to go a little further and say that they're produced by the interstitial or Leydig cells. And then finally some sperm production. And actually that's an error. It says these images are not in your text. They actually are. I'm sorry. I needed to remove that, and I didn't get that removed before I did this lecture. 65 to 75 days is about how long it takes for sperm to be produced. Each day a male makes about 300 million. That's quite a few. And as I mentioned before, they migrate to the epididymis. This is the structure of the sperm. I will not ask you the specific questions or parts, but I am going to ask you the steps in this process, and the next slide takes care of that a little bit better for us. So this is the events, and this image is also in your text. This is the image that takes you through the events or steps in spermatogenesis. So let's just walk through that for just a second. Now remember, sperm production is going to really begin once puberty kicks in. Now for the male, once they start puberty, things start changing in their bodies, and this process begins. Please be aware that it takes several years before they being the male, are actually producing viable sperm. So if a young boy starts entering puberty around 12 or 13, it might not be till he's about 15 or 16 that he can actually father children. Now, he will secrete semi-formed sperm. They will not be viable in most cases. Now, there's always the anomaly, the guy that makes it in the situation that makes it on the front of National Enquirer or something, or a 12-year-old fathered a child, but 99.9% of the population of males need a couple of years before they're actually producing viable sperm. So do keep that in mind. So what happens, though, in this process, and this is why sperm can be produced throughout the lifespan of the male. As you'll see with females, there's a limited time from onset of puberty and the beginning of the formation of the egg. And by the way, females don't need 2 years. As soon as they begin their menstrual cycle, they are producing viable eggs. Now, they may not be as fertile as, say, someone who's 17 or 18 years old. However, they are absolutely capable of becoming pregnant and bearing offspring from the moment of their first ovulation. We'll talk more about the female a little bit later, though. But with females, that process ends with menopause. So the woman hits late 40s, early 50s, and that process will come to an end. However, a male will continue to produce sperm throughout his lifespan. And the reason that is because in the process of spermatogenesis, it always begins with a mitotic division in which a diploid cell, listed here as a spermatogonium, goes through a mitotic division. It makes a copy of itself. One of those copies is saved to continue to divide again. So we have a saved spermatogonia, and you have a primary spermatocyte, which then can undergo meiosis 1. So it differentiates and goes into the process. So we save one for the next division, and one then moves forward through this process. So a primary spermatocyte that's gone through a little bit of development, differentiation, becoming a little more specific than the one that was saved. It then, as a primary spermatocyte, enters into meiosis 1 and goes through a division. And in that division in meiosis 1, it goes from diploid to haploid. So we go from a primary spermatocyte to a secondary spermatocyte, and we are now haploid. So that's the first part of meiosis. In meiosis 2, if you'll recall from your A&P1, meiosis 2 is very similar to mitosis. Those cells simply make copies of themselves. So now you have 4 viable or nearly viable sperm, they're not quite there yet, coming from that one diploid cell that was sent through the process. So you go from a primary spermatocyte, goes through meiosis 1, you'll have secondary spermatocytes, you've got 2 of them, They're haploid, and notice they're connected to each other. That's an important thing. It has what's called a cytoplasmic bridge, and they divide again, and now all 4 of them are connected by cytoplasmic bridges. They're referred to as spermatids, and it isn't until they undergo spermatogenesis, and that's important. This whole process is spermatogenesis with the T, but the division of the 4 connected cells into individual spermatozoa is called spermiogenesis. Please be careful with that. Spermiogenesis is not the same as spermatogenesis. It is a subset division. The spermatids were connected, and then they went through spermiogenesis, and we have sperm cells. These will now migrate into the lumen of the seminiferous tubule and into the epididymis to complete their maturation. And therein is actually the process of spermatogenesis. This process is certainly under the influence of hormones. As I mentioned before, FSH and testosterone are very much necessary for those divisions. These first hormones you should already be familiar with. Gonadotropin-releasing hormone is being produced in the hypothalamus. It'll stimulate the anterior pituitary to make LH and FSH. Notice LH, we now have a specific function. It targets or stimulates the interstitial cells to secrete testosterone. Now, testosterone is a lipid-soluble hormone, mainly coming from cholesterol, and it's regulated by both the LH and the GnRH. They're both involved in that regulation process. We're going to look at that in just a second. FSH, it has a more indirect effect, but remember, it's got to work together to promote sperm. And then inhibin, secreted by those Sertoli cells, will inhibit FSH. And this is a nice picture over here, and it is in your text. Sorry that that's still up there. But this feedback mechanism takes you through gonadotropin-releasing hormone to the anterior pituitary, remember, through the hypophyseal portal system, FSH and LH, with LH going to the Leydig cells to secrete testosterone. As testosterone increases, it has a negative feedback on both the anterior pituitary and the hypothalamus to regulate both GnRH and LH. The FSH that's secreted will then go working with testosterone and the nurse or Leydig cells to control the formation of sperm, and it will also secrete inhibin, which has a negative feedback on the FSH. So this is your function. Here's what testosterone does, and some cells will convert testosterone to dihydrotestosterone, or DHT, and DHT has some very specific functions in the male characteristics and also in prenatal development. And that really takes you through the functions then of the male and spermatogenesis.